So we can move to what we call peptides. So with the peptides, we have to look at this scenario. You have a local gene that is basically introducing a new protein supplement. A biology student reads the ingredients of this supplement and finds out that mostly this supplement consists of peptides that are derived from hydrolyzed proteins. So the point is that in the market or in the supermarket or whatever you are, you can find out that actually some food supplements are being sold. So as a person who has attended biology classes, you need to know the impact of these supplements. So that means that you need to basically read and understand the labels or the ingredients that are contained in these supplements and how they are supposed to be taken, but also their health impact. So here we are, we have a gym that is recommending proteinous supplements to their customers, and you have to read and understand what is in there. So if they talk about peptides being contained in these supplements, well, what should come into your mind? Of course, the peptides are coming from the proteins. So that's why we are talking about hydrolyzed proteins. So that means that if proteins are broken down, we can get peptides, and if peptides are broken down, we can get amino acids. So the peptide, how does it look like? And what is the process of formation of the peptide? And if we hydrolyze peptides, what do we get? Okay. Of course, we would expect that we have peptide bonds in these peptides, but how are peptide bonds also formed? What are the health and the risk benefits? What are the health benefits and the risks associated with these supplements? Okay, so let's just look at the content related to this and see if we shall be able to answer the questions. So let's start. So peptides are formed when you have a few amino acids basically combined. So you don't expect a peptide to be a long chain of amino acids. You can, or when you have two amino acids actually combining, then you have a dipeptide. This process, like we said before, is a condensation reaction where you will have water being released. So condensation reaction is actually reversible, meaning that it can go either way. So when we have two peptide molecules combined to form a dipeptide, it's possible that we can get we can get the peptide from this we can get peptides from this dipeptide, and that reverse reaction is what we term as hydrolysis. Okay. So the peptide is made up of a few amino acids, and these amino acids are connected or are bonded by peptide bonds. And we say that the peptide bonds are between carbon of one amino acid to nitrogen of the other amino acid, basically from the carboxyl group to the amino group. Okay, but if you have a very long chain, then we can no longer be talking about a peptide, but we can be talking about a polypeptide. So this is basically what we have here. You are having one amino acid combining with another amino acid to give us this. So the bond that has been formed is here. That is, this is the new bond that has been formed. The bond is between nitrogen and carbon. Okay, so that is a dipeptide because we are having two amino acids there. Plus water is given out because this is hydrolyzed. This is condensation. So the protein is actually very complex and can undergo a lot of folding. But a peptide, just like this is a simple representation, just a few amino acids, and we have the peptide. Okay, and then this is a very complex protein. That's what you could. So, the properties of amino acids, they shouldn't be very different. I mean, the properties of peptides should not be very, very different from the properties of amino acids. Okay, that's very important to note. So when we talk about peptides, we're talking about a few amino acids that have been formed through condensation reactions, and they are connected through what you call peptide bonds. Okay, so they can exhibit properties of amino acids and some proteins. Of course, not very complex proteins. Peptides vary in their roles. So that means that in our bodies, we actually have different peptides that actually do different roles. For example, you can have peptides that are chemical signals. We can have peptides that are basically hormones. We can have peptides that are basically enzymatic in nature. Okay. The particular peptide that we have is actually defined or is determined by it's the sequence of amino acids in that peptide, and that definitely will influence the structure and function of that peptide. Peptides can be synthesized by our bodies, or they can be made in the lab. And that is why we talked about synthetic enzyme, and that enzyme is a peptide. Okay, so we can have these peptides that are manufactured in the lab can actually be used for therapeutic purposes, and that's why it is possible that you can be given a hormone that is basically a peptide, and that hormone is basically going to treat certain conditions, and this is what we talk about when we say hormone replacement therapy. Okay, so you can be given some of the things in the hospital. So again, when we are forming the peptide, it is condensation. When we are breaking the peptide, it is hydrolysis, and it's what we are having there. Okay, so we know what a peptide is. A few amino acids form a peptide, and these amino acids are connected through peptide bonds, and these peptide bonds are basically formed when you have the amino group of one amino acid combining with the carboxyl group of another amino acid. The bond is basically between carbon and nitrogen. And therefore, if we are to illustrate how two amino acids actually form a peptide, I mean a dipeptide, two amino acids combining to form a dipeptide, what happens is that it is a condensation reaction where water is given out. Okay, and you can illustrate that where you have the amino group and the carboxyl group basically reacting. Okay, so the peptide bonds help in holding these amino acids together, but also these bonds can actually determine the extent of stability of this peptide. And like we said, these peptides can play different roles: enzymatic role, support or structure role, depending on their structures. Can you state one benefit and one potential risk factor one potential risk of consuming excessive peptides in supplements? Yes, so the peptides, peptides that you can find in these supplements can help. For example, if your body lacks some of these peptides and you take some of these supplements, they can actually help in improving your health or your nutritional status. The challenge is, or the potential risk is, it is it is not very easy to determine the extent of these peptides that you actually need. Okay, how much does your body need? So if you don't carry out the appropriate biochemical tests to determine the particular amino acids that you're missing, such that you get what your body requires, the risk is that you can get excesses of this. Okay, you can get excess of these proteins or these peptides that you are actually taking in these supplements, and that means that you are going to make your if I can say, excretory system overwork because you're pumping it with all these supplements that they need to handle and get rid of. So that's the challenge that you have.